Hello everyone, I am Dr. Vareshang Tangpu, Assistant Professor of Zoology at RIE NCRT Mysuru. I am one of the reviewers of Zoology textbook of Tamil Nadu State. It is because of that a request from my colleague Dr. S. Shamyam, the Director of SCRT Tamil Nadu. I am here to present you my dear biology teachers on the topic Parthenogenesis, which is as per your curricular requirement. Now, as we all know, parthenogenesis is a special type of reproduction which can be considered as one kind of sexual reproduction, which occurs in some plants and some animals, wherein an ovum can develop without fertilization. This phenomenon of parthenogenesis is also known as virginal development or virginal reproduction, because if we look at etymology of the word parthenogenesis, the terms came from the Greek word parthenos, meaning virgin, and genesis, meaning origin. So, all the individuals formed by parthenogenesis are called as parthenots or parthenogenes. Now, let us look at the classification of parthenogenesis. It is mainly divided into two. Number one, natural parthenogenesis or spontaneous parthenogenesis, and number two, artificial parthenogenesis or induced parthenogenesis. In the natural parthenogenesis, it can be further classified into two types, that is complete parthenogenesis, which is obligatory, meaning to say that the individuals completely reproduce by parthenogenesis process. These are found in cladocerans such as Daphnia, Platyhelminthes, rotifers, and wasp. The second type under natural parthenogenesis is called as incomplete parthenogenesis or cyclic parthenogenesis, because it alternates with the sexual reproduction along with the parthenogenesis. Therefore, it is called as incomplete or cyclic parthenogenesis, example of which are found in honeybees, aphids, and some rotifers. Now, the artificial or induced parthenogenesis are given special treatment. It could be chemical treatment, physical treatment, radiation treatments, and these are successfully carried out in sea urchin, moth, fish, frog, turkey, hen, and also in mammals such as rabbit. Now, if we further look at the classification of parthenogenesis based on the behavior of chromosomes, it can be classified into haploid parthenogenesis, diploid parthenogenesis, and the third one is apomictic parthenogenesis. It can also be classified based on the sex of the offspring, that means male or female of the parthenots. Based on that, it can be number one, arenotochi, that means only male parthenots are reproduced. Teletochi, that means only female parthenots are found in the population under this parthenogenesis. And the third one is amphitochi, meaning to say that both male and female parthenots are found under this type of parthenogenesis. Now, the first type is the haploid parthenogenesis. In this case, normal meiosis occurs and the parthenogenetic haploids develop into males. The best example comes from the honeybees. In honeybee, there is a queen which is fertilized only once by a male. The male is called drone. She stores the sperm during the mating in her seminal receptacles. There is a special type of storage system in the queen that is called the seminal receptacles, and as she lays eggs, she can either fertilize the eggs or allow them to pass unfertilized. She has a choice. In this, the honeybee, the queen, has the freedom or the choice to either fertilize the egg or to pass them down unfertilized. If they are fertilized, that means honeybee has 2N chromosome, 32 chromosomes, become the female or the worker. And if they are unfertilized, that means it is N haploid set of chromosome, that means 16 chromosomes will be found in case of male. Male drone will continuously undergo mitosis process to form the sperm, whereas the gamete that forms the matured ova can be fertilized by the sperm, forming a zygote. Then that restores the diploid set of chromosome once again. That means 32 number of chromosomes are restored. That will form into female workers. Among the female workers, only one or few can be fed with special type of food that is called royal jelly, and that becomes the queen. Now let us look at the second type of parthenogenesis, that is diploid parthenogenesis. It is also called as automictic or meiotic parthenogenesis. In this case, normal meiosis occurs, but chromosome number is doubled by a process called diplosis. This diplosis occurs either before or after meiosis. This diplosis process takes place in two methods. Number one, the diplosis by autofertilization. That means during oogenesis, meiosis 1 is normal with porocyte and meiosis 2 is suppressed. Second, porocyte fuses with egg nucleus to produce diploid set of chromosomes. The best example is exhibited by aphids. The second type of diplosis is by restitution, which is found in some insects. That means there is karyokinesis but no cytokinesis. The chromosomes of both nuclei get arranged on the equator and proceed for M2 to form the diploid oocyte and secondary polocytes. The best example of this is exhibited by Hymenolipis and Lepidoptera. Now further elaboration of the diploid parthenogenesis in aphids can be shown diagrammatically in this slide. During summer, the female undergoes parthenogenetic process of reproduction only. It can go up to 10 generations. So parthenogenetically, the female will have 2n number of chromosomes, diploid set of chromosomes. So after about 10 generations that pass by, that will come along with the autumn season, the female will become the stem mother. That is a parthenogenetic female which can produce two types of progeny parthenogenetically. One becomes the male producer and the other becomes the female producer. The female producer will continue to produce a functional ova which is 2n number because of the diplosis, doubling of the chromosome, that continue to retain 2n number of chromosomes in meiosis, and the female sexual form finally will undergo meiosis process. So thereby each of the gamete will have n number of chromosome, that is a functional ovum. Now the male producer will form 2n minus 1 set of chromosome. That means they become the male sexual form. That means it will lack one of the sex chromosome. So 2n minus 1 number of chromosome, which undergoes meiosis, that results in n set of chromosome, that becomes a functional sperm. The other one, n minus 1 set of haploid set of chromosome, they will die off. Therefore, the functional sperm and ovum will fuse, undergo a zygote formation during spring. Therefore, in the spring, they will continue to undergo offspring formation. So in the summer, continue to produce parthenogenetically, only female population will be found. Whereas in the winter, the aphids will produce both the male and female aphids. Now let us look at the third type of parthenogenesis, which is called as amictic or apomictic parthenogenesis. In this case, no meiosis occurs. Apomeiosis means meiosis is suppressed or there is imperfect meiosis. The best example comes from the shrimps and Daphnia. In this case, the Euconia, having 2n number of chromosomes, which is supposed to undergo meiosis process, in meiosis 1 is suppressed, thereby resulting in 2n number of chromosomes of the oocyte. Then M2 will be formed, that is a normal mitosis, which will form 2n number of chromosomes. So this continue to grow, and diploid female will continue to have 2n number of chromosomes. In this amictic or apomictic parthenogenesis, only female population will be formed. Now the specific example comes from the Daphnia. In Daphnia, there are 12 to 20 number of chromosomes. Under favorable condition, they prefer to reproduce parthenogenetically. That means only female population will be produced. Parthenogenetic daughters are formed from the parent. Only under unfavorable conditions, that means lack of food or predators and other kinds of disturbance in the environment, parthenogenetic son, male, will be produced, and it will have haploid set of chromosomes. Whereas in the female, they will have 2n number of chromosomes. Now based on the sex of the offspring, they can be classified into arenotochi, where only male parthenoids are produced. The best example comes from the honeybee. The second type is the teletochi, where only female parthenoids are produced. The best example is found in aphids during summer, as we have seen. And the third type is amphitochi, where both male and female parthenoids are produced. The best example comes from the aphids again in a cyclic parthenogenesis during winter. Both the male and female parthenoids are produced. Now, that's all about the natural parthenogenesis. Let us look at the artificial parthenogenesis, the development of a ripe egg by certain treatments but without fertilization is called artificial parthenogenesis. It is one of the great interests among the biologists as it throws light on the activation of egg, including humans' ovum, by the sperm at the time of fertilization. It is best described to occur in lower groups of organisms such as annelids, mollusks, echinoderms, amphibians, aves, and also mammals. The best experimented success come from the sea urchin, moth, fish, frog, turkey, hen, and rabbit. So this artificial parthenogenesis by certain treatments come from chemical, physical, and also treatment with radiations. Under chemical treatment, the most successful result comes from sea urchin. So the sea urchin eggs can be treated with hypotonic solution or hypertonic solution, or they can be treated with chloride salts of potassium, calcium, sodium, magnesium, and lithium. Even the mature egg can be treated with weak organic acid such as lactic acid, oleic acid, and fatty acids. In the physical treatment, the matured eggs can be treated by giving temperature shock. That means by transferring the egg for a short period of time to a very cold temperature, or they can be treated at a warmer temperature, that is above 32 degrees Celsius. They can also be given electric shock, or the eggs can be shared in ordinary seawater, or you can give a prick. The egg can be given a prick with a fine glass needle. So the treatment with radiations. An ovum can be fertilized by genetically inactivated sperms having treated with UV light. Now, let us look at the advantages of this process of parthenogenesis. Number 1, means of reproduction. That means parthenogenesis is the simple method which ensures continuity of the race. Number 2, rapid multiplication. A large number of individuals can be reproduced within a short span of time, such as in aphids. Number 3, elimination of disadvantageous characters. That means parthenoids with unfavorable mutation are eliminated altogether from the population, as there is no meiosis nor amphimixis. Number 4, persistence of advantageous characters. Sometimes useful characters appearing in the parthenoids are retained, as there is no genetic segregation. Number 5, sex determination. The sex of the individual offspring is determined by the number of chromosomes. Therefore, parthenogenesis is a means of sex determination. Number 6, avoidance of polyploidy. Parthenogenesis prevents polyploidy due to the absence of meiosis. Number 7, fertility. Parthenogenesis prevents sterility in the races, thereby promoting fertility. Number 8, prevention of wastage of energy. Mating during sexual reproduction requires a lot of risk, time, and energy. Parthenogenesis does not involve mating, so most energy can be used for feeding and other vital activities. Let us also look at the disadvantages of parthenogenesis. The main evolutionary disadvantages of parthenogenesis is that it tends to eliminate variations in a population. The offspring developing are more alike to and mostly the photocopies of the parents, whereas a sexually developed offspring exhibits marked differences from the parents. I hope this video suffices the requirement of your curriculum. Thank you so much. Thank you.